Hi everyone. We're going to be wrapping up the very end of chapter 12, which is on just finishing up the different applications for DNA technology. Okay, so the question is, how can we identify genetic differences between human beings? Clearly, all of our genomes are different from each other, and that's what makes us different, like different people. But how can we see that genetically? Well, we've talked about one, which is short tandem repeats. And remember, that's just segments of DNA that's repeated multiple times, and we tend to have different numbers of repeats, and so that's one way we can tell that we're different from each other. But there's also something called single nucleotide polymorphisms. Single nucleotide polymorphism is a single base pair variation in the genome. Um, and so an example here is that three different individuals, there will be a specific place where there just is a higher frequency where we have variations in the base pair that exists in that one location. And so these single polymorphisms, single nucleotide polymorphisms can be found in both coding regions, that means segments of genes that lead to proteins, and it can also be found in non-coding regions of the genome. The reason why these are so important, you might be thinking like, well, if it's just one nucleotide that's different, like, is it really that significant? Well, it is because when you have this one base pair that's different, it can actually change the restriction site because it's changing the frequency, the sequence. And so where a restriction enzyme would normally cut may be different now between individuals because of that one nucleotide difference. And so when you change the restriction sites, you're going to be changing the length of those restriction fragments. And these differing lengths of restriction fragments leads to something called restriction fragment length polymorphism, RFLP. And this is, we can use RFLP to be able to determine differences between human beings. So here is just a visual depiction of what we talked about, where these red lines indicate where there's one nucleotide difference between all of these different individuals. And you can see, even though it's just very minute differences, just a few nucleotides, the restriction enzyme now cuts at different places. So now it cuts in these different locations, leading to these very different looking patterns of DNA fragments when you run it through the gel electrophoresis. And so this is another way that you could identify human beings and see, like, for example, in crime scenes, you can determine which person matches up with the suspect data. So other uses for this, it's a genetic marker, which just means it's like kind of like your genetic fingerprint, and you can tell who's who. But you can also use RFLPs to identify disease-causing alleles. So this is just another way where you can look and see, like, some people who tend to have this kind of fragmentation due to their single nucleotide polymorphisms and SNPs. You could say, well, that one nucleotide seems to be really causing this kind of disease, and do more research onto it. Okay. So genomics is a study of just an entire set of genes. And so there was this huge push in biology to start to sequence. Once we started learning about all these different tools, biologists wanted to sequence the whole genome, figure out what is the entire code of all these different organisms. Now, you might be wondering what the purpose of that is. For one, you get an insight into the evolutionary relationships between different organisms. And so you can compare the gene sequences between organisms and see which ones are more similar and which ones are not. And that might give you a clue of who the common ancestor was. The other thing is, like, when you really just understand genes, it gives you a better understanding of what those genes do. And so once you establish that relationship, you can start to understand things like diseases, what happens when those genes are not in the right sequence like they should be. And so it helps us understand so many different things in biology. So a huge project that was a manifestation of genomics is the Human Genome Project. And this took 13 years to complete. And this was the push to sequence the whole entire human genome. So they found some very interesting things when they did this. They found that only 1.5%, so very, very minute amount of DNA actually codes for what we would consider useful things like proteins, tRNAs, and RNAs. So you might be wondering, like, what does the rest of that DNA code for, the 98.5%? Well, the rest of the DNA, there's a big segment of it is just introns and regulatory sequences. So remember, introns are just those parts that are inside a coding sequence but don't actually code for anything. Regulatory sequences are like promoters, enhancers, microRNA. So these are all just different things that actually help regulate the DNA. So this makes up about almost 25% of the genome. But a huge chunk of it is even more random. It's non-coding DNA that really doesn't seem to have that much use. And we've been talking about these repetitive DNA sequences that don't seem to do much. We talked about short tandem repeats. There's also something called telomeres. And telomeres are these short repetitive sequences of DNA that are kind of like the caps of chromosomes. And so scientists have found that they think that these telomeres actually are protect chromosomes from unraveling. And so even though it doesn't actually code for anything, you could think of it as like extra bubble wrap that kind of caps like the end to keep it keep it staying together. They've also noticed that a lot of these repetitive DNA sequences are associated with transposable elements. So transposable elements are just DNA segments that can kind of jump around and almost move from one place to another. Now, just remember, like, that's not necessarily really a good thing. If you think back to the section on chromosomes, if sections of chromosomes move from one place to another, it might lead to cancer because that segment of DNA is now coded with a different promoter. So, so some people think that a lot of this random DNA is just the byproduct of a lot of viruses that have inserted their DNA into our human sequences. And they're still trying to figure out the purpose of these repetitive sequences of DNA. All right, guys, thank you so much for watching. Bye.